on campus or off campus, so you would be able to see those slides. Okay, but for example, if we cover something on the board or something, so it may not be that efficient for the students who are not on campus or who are not in class. Okay, I think this is what I understand by hybrid mode. So if someone want to add something that how would it work out and whether it's working out well for other classes. So let's hear from you guys now, one by one. Abhilasha, you want to say something?
Sir, I'm at campus, so I think it's
So
fine for me.
now you actually can lower your hand. So whosoever raised, now you all can lower your hand. So let's discuss this point for next few minutes.
Sir, maybe you can keep laptop in the class while you're teaching and stream live on the Zoom, just like right now, but you can use the board.
Yeah, but then probably whatever I write on the board, so that will not be visible to students who are not on campus or in the class, right? This is what I was saying.
Sir, I am saying that while students are in class,
yeah,
you can also keep a laptop with camera on towards
okay.
the board, and then people can see, because NGP sir uses a board for BIO404 and it's fairly visible.
It's fairly visible. Okay. Okay.
So it's basically that you can shift from board to slides whenever you want. For example, right now, if you are showing your video, so instead of that, there will be a video of the green board along with the slides. So you can share screen and you can
okay, so
board.
Okay, so I think probably from this share option, so we might have to put something. No, it cannot be the whiteboard, right? Or it will be unshare. See, it with whiteboard basically, so you'll have a blank board on your screen, and I can write something on that whiteboard using whatever the thing that we have, right? But for example, if I'm using that classroom board, so would that also be visible?
Sir, so I think what is needed is, because the slides will anyway be provided, what is needed is that if you're writing something on the board and you're following the lecture, then the student should at least know which slide you're talking about. That's it, because they will have the slides anyway. So it could be somewhere like the pane of the camera that's on you gets shifted a little bit so that they know which slide you are talking about. That's all. Otherwise, the focus should remain on the board itself.
Which board? Okay.
The board that you're writing on in the class.
In the classroom board.
Yeah, sir. I think this can work.
Okay, so what we'll do is let's try it out for this Thursday's class. Okay. Until Thursday, so we will do the online mode on Thursday. So we'll try one class. Maybe we don't cover the next topic. So try to answer your question and try to, for example, if you have something that we can try to explain using board, and if it is working fine, so we'll continue with the hybrid mode, right? Otherwise, we might shift on to the this online mode, unless you all are here. So would that work?
Sir, is it compulsory for students on campus to be in class, or can they also attend online?
Well, for example, if you have some problems, so I don't know, so if you are quarantined in quarantine visitor hostel, probably you don't need to come, right? And obviously, when you come to class, so you have to wear all those masks, and you have to be spread out in the whole lecture hall, right? So lecture hall number 6 is quite big. So basically you can spread out. But again, question would be whether that same screen, that board, will be visible in the end. Okay, so whosoever is sitting in the back of the class, so whether they would be able to see anything written on the board. So again, that's going to be a problem, right? Well, I don't see a better way of dealing with this problem, unless all of you are here and you don't have any COVID protocol to follow, right? Because then all of you can, because we are only 90, right, in this batch, approximately 90. So 1/2 of the class will be covered in that case, and then you can reasonably well see whatever is written on the board. And obviously slides will be displayed because these two display screens are there, right? And if something needs to be explained, that can be done using the central green board. Well, think about it. Think among yourself, discuss among yourself for next 2 days, and let's try it out for Thursday. And what we'll do is we don't cover the next topic. Okay, so whatever we have done until Wednesday, okay, so we will try to talk about that only, right? So because at least whatever we are talking about now, so all of you are actually grasping it uniformly, in the sense the whatever is available, it's available uniformly to everybody. Okay, but let's try it out for one of these classes on Thursday. How is it working out, right? Does that sound a good plan or what?
Yes, sir.
Fine, sir.
Okay, so that's what we are going to do. Now, let's quickly talk about if you have any doubts. So one of the question I see is, in one of the slide, it was mentioned that antigen interacts with BCR, and in turn, antigen is processed and presented to cells, and then T cells release cytokine. Yeah, I understand what you're saying. Now, question was, is this process only associated with T helper cells? Well, essentially, yes. Okay, because those cytokines produced by T helper cells are going to act on your cytotoxic T cells also. Okay, but obviously this process, because B cells, if they take up antigen through BCR by the process of this internalization of BCR bound to antigen, right? They will also be processed through this mechanism that we call as the internal processing or exogenous antigen processing, and then you can actually load those antigen on MSC class 2 for sure. But there is one pathway that we call as cross antigen presentation, where this internalized antigen will be processed and loaded. The peptide generated will be loaded onto MSC class 1 to activate your cytotoxic T cells. Okay, but you also have to understand that initially during a response or immunization, so you don't have many antigen specific B cell receptor expressing cells. So there are on an average, so these cells actually range from 500 to 1000 for any particular epitope, right? Sometimes it's even less, but 1000 if I'm saying, so this is actually on the higher side. But after a primary response, so then a lot of these B cells get expanded specific to that particular antigen, right? And in memory response, you are left with so many of B cells, which actually can facilitate this process. um, during a secondary response. So B cells are good antigen presenting cells during a secondary response, but not playing a major role during the primary response. Okay. Yeah. So then if yes, then how does cytotoxic T cells activate and function? Well, cytotoxic T cells are predominantly engaged by the other two types of APCs, macrophages and dendritic cells. Okay. Let's consider epitope X interacts with BCR. The processed antigen presented by B cells to T cells need to be necessarily the same epitope. Well, you have to understand, you have the whole antigen, right? A lot of epitopes are present, and maybe one of those exposed epitope present on surface of this antigen will be recognized by your antibody, and this whole complex is internalized. Now, whole complex means the whole protein actually has all kind of T cell epitope and B cell epitopes, and they will all be processed in that fashion, right? So it doesn't have to be exactly the same epitope that actually binds to BCR, and it will be processed and eventually generate to be loaded onto MHC class 1 or class 2 to activate T helper or cytotoxic T cells. No, it doesn't have to be. Things will be much more clear when we talk about antigen presentation. Okay. But for the time being, remember, B cells play a major role as antigen presenting cells during a secondary response, not during primary response, for the reason that you don't have sufficient number of B cells specific to any given antigen. And with with secondary response, so you have a significant number of these cells. That's the answer. Okay, any other question? If not, so let's talk about the next topic.
Sir,
Yes.
sir, a short question. So we have talked about MHC class 1 and 2 like in multiple lectures. So could you like summarize and then briefly talk about like so the general features of MHC class 1 and 2 to just to sum up, like what are the major roles played by them? So
Well, do you want me to talk about antigen processing and presentation now?
no, sir. I mean,
Okay, just a summary.
Yeah. So
Okay.
like
All right. Yeah, I understand. So there are predominantly two pathways of antigen presentation. Okay. One is where basically the antigen is present inside these cells, and that is because of the intracellular infections. Okay, so these viruses, parasites, which can be internalized by these cells and they replicate inside these cells, right? Now, you have the proteasomal machinery. Proteasomal machinery is going to degrade those big antigens, generate those small epitopes. Okay, those small epitopes are loaded onto MSC class 1, okay, predominantly, and they are going to activate your cytotoxic T cells, antigen specific CTLs. That's one. Now, if the antigen is internalized through the process of phagocytosis, okay, so then this will first get into this phagolysosome, right, or endolysosome, if the antigen is soluble or particulated matter. So that will decide which pathway it will take up, or through receptor mediated endocytosis, right? Now, the antigen was initially present outside. Now this is present inside these cells. Now, whatever the peptides will be generated from those proteins in this antigen, so they are predominantly loaded onto MSC class 2. Okay, this is going to engage your T helper cells. Okay, this is called exogenous pathway of antigen presentation. The earlier was the endogenous pathway of antigen presentation. Then you also have the third pathway that we call as cross antigen presentation. In that, what happens is you have the antigen which is external, exogenous antigen, which is internalized. Okay, but the MSC class 1 peptide generated can be loaded onto MSC class 1 and they can activate cytotoxic T cells. So this is called cross antigen presentation, rather antigen cross presentation, not cross antigen presentation, antigen cross presentation. Normally it was supposed to be loaded onto MSC class 2, but now this is loaded onto MSC class 1 to activate your cytotoxic T cells. This is in brief. These three pathways, we are going to talk about the details of these three pathways. The purpose is to activate your either T helper cells or cytotoxic T cells. B cells, they can be activated without any antigen processing. Okay, because they can actually see these native antigens through their BCR, right? And they can get activated. Nonetheless, for efficient activation of B cells, you require help from CD4 T cells, predominantly through the cytokine produced and some of the surface molecules like CD28 and its counter ligand. So this is broadly how these effectors are brought about to activate either your cytotoxic T cells or your T helper cells. And you know that well-helped cytotoxic T cells control any intracellular infection or cancerous antigen much better than the cells which have not received this help at the time of their induction. Okay. Does that make sense?
Yes, sir. Yeah, thank you.
Okay, can we discuss the someone is willing to discuss the mid-sem paper? Well, ask if we start talk well very broadly, so maybe I can talk about. So what was the first question? So if you have it handy. Abhilasha, so what you want to discuss from the mid-sem exam? Any question?
No, sir, it's fine for me.
Okay. Mridula, so can you turn on your this speaker and say maybe the last question? What was the last question? So where basically these different types of cells were to be named, right? Yeah. So who has it handy?
Sir, I have it.
Okay, now say it. So and there were eight different types.
Yes, sir.
What was the first one?
The first one is macrophage in liver.
Yeah. So basically these are Kupffer cells, right? Second,
Macrophage in brain.
microglial cells.
And, CD11B positive and CD11C positive.
Dendritic cells.
Both one is modified epithelial cells in gut involved in facilitating immune induction.
M cells.
Then CD3 positive and CD4 negative.
Cytotoxic T cells.
The cells which show an extended network of ER upon antigen presentation.
All these are activated B cells or plasma cells.
The cell type involved in ADCC.
NK cells.
Last one is cell type that contain histamine and critical
Mast
fiber.
cells.
Yeah.
Okay. All right. So I think let's whatever 20-30 minutes we have. So let's talk about something. As a new topic. Okay. Okay. So we were talking about the antibodies, and so this slide actually talked about blocking and signaling antibodies. So how do you differentiate between blocking and signaling antibodies? And broadly. So for example, an antibody can be called as blocking antibody if it binds to the ligand, so that ligand is not available to activate the receptor. So this is going to be serving as a blocking antibody. The second aspect is basically it binds to the site from outside so that this ligand is not able to bind to it. That's second. Third one is because there are multiple receptors which need to form either dimer or trimer, and if an antibody can stop the interaction of different units of the receptor, this will also serve as a blocking antibody. And fourth one actually is showing that somehow if this surface receptor has to attain certain configuration to start inducing signaling, and the antibody is stopping that, so this will also be called as a blocking antibody. And in fifth one, actually it is shown that this antibody is binding to exactly the same site where your ligand is binding. And in this case, you can get away with having ligand to stimulate this receptor, and your antibody actually can stimulate the receptor in this case. So this would be called as signaling antibody, right? This is what we discussed earlier. The next thing is we are going to talk about effector function associated with antibody. Okay, so we know that antibodies, they generally do not need to kill. Rather, they don't actually kill or remove a pathogen solely by binding to it. But then how basically they bring about these functions? Okay, those, see, binding is one thing. So you have the antigen, antibody is coming, it's binding to it. Okay, this is not necessarily killing this this pathogen or cancerous antigen just by that, just by doing this function, binding to it, right? Sometime it can. Okay, for example, if antibody is bound to it, right, you actually have generated an immune complex of antigen and antibody. They are actually in complex, and then you can activate complement system, and then with this complement actually is going to activate on surface of, for example, this virus, bacteria, or whatever, and then it can bring about that killing function. Okay, the other thing is how is it going to help? For example, if antigen is sequestered, if it is bound, and for example, if this is a replicating unit, let's say if this is a virus and the antibody has already bound to it, so it will basically prevent the internalization of this virus or parasite in the cell. And you have to understand that one virus or one bacteria or one whatever is not doing much. So unless it gets into these cells and make millions and billions of copies, and then you start developing all these diseases. And what your antibody have done actually is they have actually bound to this incoming virus, and they have stopped its internalization and stopped the replication step, right? So that is one of the function of, this is called neutralization. These antibodies can bind to these microbes, and they can neutralize their subsequent step for replication, and so on and so forth, right? Nonetheless, there are also some effector molecules associated with these antibodies. Okay, and those are the one that we are going to talk about in today's lecture. Okay, so understand the V region, the variable region. Okay, so you have this antibody molecule, so which has actually the this projected arms and the stalk, right? The stalk is Fc portion, and these are basically the variable region. Okay, the variable region is binding to antigen, and the constant region, particularly the CS region, is responsible for a variety of collaborative interactions, okay, which involves many protein cells, and if this reaction is taking place, for example, in extracellular spaces, tissues, right, that result in the effector function of an antibody molecules. Now, what are these? Now, take-home message from this statement is the CH region is responsible for the effector function of antibody molecule, right? So these are the effector functions. One of them is opsonization, which is promoted by antibody. Second is activation of complement system. Third is antibody-dependent cell-mediated cytotoxicity. And fourth is because these antibodies, for example, are produced at one location and they have to actually traverse through these membranes or epithelial layers, get into the gut where microbe might be present in order to neutralize it. Okay, so this process of transcytosis is also the effector function of antibody molecules. So there are four. First one is opsonization. What opsonization is, I will talk about that. Activation of complement system. Antibody-dependent cell-mediated cytotoxicity, and we know that NK cells are very critically involved in that. And fourth one is the transport of these antibody molecules from the site of their secretion to the site of their effector function. For example, through crossing the epithelial layers in gut. Okay, so these are the effector functions of an antibody molecule. All right, so what is shown here is basically, for example, you have this microbe, okay, in extracellular spaces, and you have these macrophages. Normally, macrophages can take up all these big microbes, right? So by this process of phagocytosis. But antibodies, by binding, for example, let's say if you have these antibodies generated against these microbes, antibodies can bind to those antigens present on the microbes, right? And in so doing, so they facilitate, they promote the process of phagocytosis. Okay, the process by which this antibody is binding to these different antigenic moieties present on this microbe is called opsonization. Okay, you have this microbe. It has these different antigens. If your antibody is specific to those antigens, so it will go there, it will bind to it. And then by involving this constant region, the constant region of antibody molecules, it will facilitate its internalization. Okay, as it basically written in this box, antibody promotes opsonization through binding to Fc receptor on phagocytes. And you know that these Fc receptors are present in large abundance on surface of these innate immune cells, particularly macrophages, also NK cells. Okay, so the process of coating of this microbe with antibody, followed by its internalization involving Fc receptors, and Fc receptor is going to bind to the Fc portion of the antibody molecule. And in so doing, so this whole process is facilitated and this microbe is internalized efficiently by your macrophage, the cells of the innate immune response, right? And eventually this microbe is cut into these pieces. So basically you have killed this microbe by different mechanisms. One of them is production of reactive oxygen species, enzymatic activity that you have in phagolysosome and so on and so forth, right? So this process is called opsonization. That is promoted by Fc portion of the antibody molecule. Second, so you have this bacterium, right? Bacteria is, for example, bound to this antibody. Okay, now if you generate this antigen antibody complexes, this is antigen, this is antibody, so you have basically brought about this interaction. So you have generated what we call as immune complexes. Immune complex is what? Where you have antigen bound to an antibody. Now, once that happens, so this interaction, this formation of this immune complex actually can activate complement system. And once complement is activated, so the complement activation will take place on the solid matrix where antibody is bound. And antibody in this case, for example, is bound to on the surface of this microbe, whether it's a virus, bacteria, or parasite, right? Now, following, so there is one step, the final step in complement activation, where we call as actually the generation of this MAC, membrane attack complex. Once this membrane attack complex is generated, so this actually will make pore through this microbe. Once those pores are generated, so actually then water can gush out very easily. And because of a lot of accumulation of this water, these cells are going to burst open. And in this case, these are microbial cells. So basically they can be controlled by this mechanism. But remember, so if you are controlling something through complement system, so this is a highly inflammatory process. And many a time, you actually even fail to control that inflammatory process, and that is more damaging than the microbe itself. Okay. But nonetheless, this is one of the other effector function associated with antibody molecule. Just read whatever is written in this box. Antibody activates complement mediated cytolysis or promote opsonization through binding to C receptors on phagocytes. Yeah, so I said in this case, it's a direct binding of antigen exposed on surface of this microbe with antibody. Okay, this is basically simply a coating of the microbe with antibodies. This is called opsonization. Okay, now through activation of this complement system, so you also generate certain compartments, okay, which we call as complement components. And those complement components can also bind to microbes. And because the complement receptors are present on your innate immune cells, okay, so that can also promote phagocytosis. Okay, the components of complement system which promote or which actually bind to the surface of this microbe and help internalization are called opsonins. Okay. All right. So the next function associated with antibodies is that we call as antibody dependent cell-mediated cytotoxicity. Okay. And these are the mediators: lytic enzymes, perforin, TNF-alpha, granzymes. All these molecules are associated with killing activity of your cells. They are produced by your cytotoxic T cells. But initially, so this function is carried out predominantly by your NK cells. Okay. Now, there are other cells also. So for example, neutrophils, you know that, so these are polymorpho nucleus cells, which actually have these preformed granules. They make these granzymes and other molecules. And they also have this Fc receptors. They also have this Fc receptor, eosinophils. They also have this Fc receptors, right? Macrophage also have these Fc receptors. Okay, and Fc receptors is one of the major reasons why NK cells get activated, right? So there are different types of those, CD16, CD32, CD56. So many kind of receptors are there. Okay, but essentially what is basically shown here in this slide is, for example, if you have a target cell has antigens on their surface, and these antigens could come from either from this target cell itself or from the infection of this target cell, right? Also, what can happen is, for example, if this is normal cell and this is transformed, because of transformation, some neoantigens are generated, particularly during cancers, and those neoantigens can bound to these antibodies on one side, right? And these antibodies, because they have this Fc portion, so they can bind to Fc receptor, right? And in so doing, so they can actually activate your NK cells or other innate immune cells. Okay, and when these cells are activated, they are going to release the cargo present in these cells. And when this cargo is released, so this is going to either make pore in the membrane of this target cell, and then the rest of the molecule, for example, perforins, they are particularly involved in making ports in the membrane. Okay, then all these lytic enzymes, particularly the granzymes, serpines, and many more, TNF alpha, so they are then basically internalized within these cells. And once they are internalized, they cause apoptosis of these cells. Okay, and with the apoptosis, with the demise of these cells, if this was an infected cell, so you are going to also control to some extent the infection. If this is a cancerous cell, so you are going to actually control the carcinogenesis. Okay, so for example, if you have one cancer cell, you have killed it, so that means actually you are trying to prevent development of cancerous conditions, right? This process is called antibody dependent cell mediated cytotoxicity. One more thing that you have to understand is that all these molecules are predominantly present in NK cells, but to some extent, for example, in neutrophils, they have many of these lytic enzymes, but not many other things, right? So there is a gradation to what extent these different innate immune cells have these effector molecules. Okay, NK cells have a lot of them, and that's why they are so good in causing antibody-dependent cell-mediated cytotoxicity. Questions on this slide? So essentially what I'm trying to say here is antibody can bind the facilitation of the antigen-antibody interaction, and eventually the function is brought about the tail of the antibody molecule that we call as FC portion. FC bind to FC receptor present on these innate immune cells, and in so doing, so they are basically inducing release of the cargo in these cells, and your cancer cell, infected cell is killed. This is called antibody dependent cell mediated cytotoxicity. Okay, transcytosis. This is basically the movement of antibody across epithelial layer. And you know that delivery of, for example, this particular class of antibody molecule that we call as IgA, because IgA actually is a mucosal antibody. So this is actually predominantly present, for example, in your tears, in gut, in urogenital tract. So wherever you have the mucosal sites, IgA is the predominant antibody isotype. Okay. Delivery of this IgA to mucosal surfaces, okay, of either respiratory, gastrointestinal, or urogenital tract, or its export actually in the milk, breast milk. So this is facilitated by the FC portion in these IgA antibodies. Okay. And once these antibodies of IgA type are present there, so they carry out their functions. I think somebody else is logging on. So we'll continue from here. Okay. So now we are actually going to stop.